So we're going to talk a little about that Mendel experiment again. So he took that purebred purple flower and he crossed it with a purebred white flower. And as we discussed, this one is going to be getting the capital letters because it's the dominant one, and this one's going to be getting the small letters because it is the recessive allele. So just to recap what we've already heard is that when he bred these two things together, he found that the only type of flower that they made was purple. And it wasn't till the generation after that, then he saw about 25% of those flowers were white again. So we're going to figure out why that was the case. So again, so when he bred that white flower, which is going to be little p, and he bred it with that purebred purple flower, big P, what he got was, we got big P, little p. So we took this one right here and we pulled it into here and it becomes big P, and this one comes down into here, little p. Again, this one comes over here and this one comes down into here. So this one that come over here is big P again, and this one is little p. This one comes into here, big P, and this one comes all the way back down into here, right here, and it's little p. And same thing for here, big P, little p. So they're all big P, little p. Since they all have a dominant allele, and this one's the recessive, only the dominant allele gets expressed. So the dominant one turns into your phenotype. Remember, the phenotype is the trait that you can actually see. This thing right here is recessive. This plant has the genetics for white flower, but it doesn't express the genetics for white flower. It doesn't show it in its phenotype. It has the genes for white flower, but it doesn't express it. Okay, I want to remember that genes are like blueprints. This has the blueprints to make a purple flower and has the blueprints to make a white flower. However, it chose not to use the blueprints to make the white flower. It only used the blueprints to make the purple flower. But now we're going to take a look at what happens if you took that next generation, that one that had a big P and a little p and another big P and a little p. So they look purple, but they have the genetics to make a white flower. So again, this is that next generation, and how we do this is we pull in that big P right there, so it gets a big P, and this one comes down into here and gets another big P. This one comes over here, big P. This one comes down here and it's a little p. This one comes over here and it's a little p. Now we typically write the capital letter first. So this one comes down into here, big P. So big P, little p. And this is why we get 75% are purple. And then this one comes here, little p. And this one comes over here, little p as well. So this is why we have 75% are purple, because of these ones right here, and 25% are white. Okay? And that's why he always saw that 75 to 20% kind of that split right there. And we don't have to draw like the actual purple flowers. He just was like big P, little p, big P, little p. And when you cross these things, there's 4 different possible outcomes for this particular phenotype. We can have big P, so that'd be a purple flower. Big P, little p, That would also be a purple flower. Big P, purple flower, and then little p, and this is your white flower. So that's 1 out of 4 possibilities, or in other words, 25% is going to be white. Now, we can call these things homozygous or heterozygous. Homozygous has an organism that has 2 of the same alleles. So big P, that would be homozygous. Or little p, that would also be homozygous. These have the same alleles, big P and big P. These have the same alleles, little p and little p. Heterozygous means it has 2 different alleles, big P, little p. So in science, homo means the same and hetero means different. So, which one is this? Hopefully you got it. That one's homozygous. Okay, which one is this one? Big P, little p. This is heterozygous. Okay, even though these 2 flowers look identical, okay, they're both purple flowers, and visually you would not be able to tell the difference between them, but genetically this one has 2 dominant alleles, and genetically this one has a dominant and recessive allele. This one is also homozygous, but it happens to be 2 recessive alleles. And how do we distinguish this one and this one right here? Well, we would actually call this one right here homozygous, which means same alleles, dominant. So it's homozygous dominant. It has 2 of the same alleles, and they're both dominant one. This one right here is homozygous recessive. This one is just heterozygous. So again, homozygous dominant, 2 of the same alleles, okay, and they're also dominant. Homozygous recessive, 2 of the same allele, but they happen to be recessive. And then heterozygous. Now, again, a phenotype just means how the organism looks. So even though this was homozygous dominant and heterozygous, they both have exactly the same phenotype. You wouldn't be able to tell by looking at a flower if it is homozygous or heterozygous, or sorry, homozygous dominant or heterozygous. However, if it's white, you know that it's going to be homozygous recessive. Now, here's something that we can try and do is like, first, can you roll your tongue? Like this person right here, like the picture. So rolling a tongue, that trait is dominant. So what we're going to do is we're going to make what is called a Punnett square. So making that box is called a Punnett square. We're going to make that a box and figure out if this male who can roll their tongue and this female who cannot roll their tongue, if they had kids together, would the kids be able to roll their tongue and not roll their tongue? So, so we use the letter R to signify for rolling. And it doesn't matter where you put your letters, but we'll put the male right here. And so the male is heterozygous, can roll their tongue, and so that person can roll their tongue, and when you look at them, you wouldn't know if they're homozygous dominant or or heterozygous. But let's just pretend that we know, and we know that they are heterozygous, so they can roll their tongue. The female cannot roll her tongue. So they are little r, little r. Now we're going to figure out what would their kids, can their kids roll their tongue or not. So again, the big R comes down, and then the little r comes over here. Okay, so big R, little r. Again, for the next one down, the big R comes down, and then this little r goes into here. So it's big R, little r. Now this one comes down, it's a little r, and this r right here comes over here, and it's also a little r. Same thing for this one right here, little r, little r. So 2 out of 4 is going to be big R, little r, and 2 out of 4 is going to be little r, little r. Or in other words, 50%, 2 out of 4 can roll tongue. And then 2 out of 4 can't. Okay. Can you fill in these Punnett squares right here? And hopefully that makes sense about how to make and fill in a Punnett square and how to read the Punnett square. So we'll do this one really quickly. It doesn't matter which individual you put on the top or on the side. So I put the male on the top here and the female on the side, but I will always switch that up. So big R, little r, big R, little r, big R, little r. So in other words, here's a male who can roll their tongue, a female who cannot roll their tongue, but 100% of the kids can roll tongue. Now we're going to put the male right here, little r, female right here, big R, little r. Okay, fill in our table. Again, 2 out of 4, or 50%, can roll their tongue. 50% can't. Okay, so hopefully that makes sense.